If you have a question, all you need to do is just ask, or you can also hit that little raise hand icon, and that will alert me that somebody's asking a question. So you can use your microphone, turn on your microphone and ask the question, or you can type the question. Okay, so I'm going to get started with the integumentary system. And the first about the integumentary system is going to be that the major organ would be the skin. The skin is divided into two regions, the epidermis, which is the superficial region, and the dermis, which is deep to the epidermis. And it is a minor region compared to, in size-wise, compared to the dermis. So let me go ahead and go for a picture. Now, the hypodermis, which is a layer of fat tissue below the dermis, also called the subcutaneous region. This is not, technically speaking, considered a part of the skin. Having said that, it is always depicted in pictures of the skin, and so we always mention it, even though technically speaking is not part of the skin. Okay, so I just want to point that out because it will be there in pretty much every picture. So down here will be the hypodermis layer, which is made of adipose tissue, fat tissue. It's also called the superficial fascia. And again, just to reiterate, not considered a part of the skin, but it's always there, so it's mentioned. The skin itself is divided into two regions. The epidermis, with the outermost region. Below the epidermis is the dermis. Now, if you look at this picture, notice how thin the epidermis is in contrast with the dermis. So the thicker part of the skin is the dermal region, the dermis. The epidermis, the outermost region, is actually quite thin, as you can see here in this picture. Both layers are made of different tissues. The epidermis is made of epithelial tissue, and we'll go into more detail later on. Whereas the dermis is made of connective tissue, a couple of different types of connective tissue. Okay. Also notice one last thing, that the dermis, the dermal layer, is where most of the stuff that the skin has is located. So receptors right here, and what else? Receptors, the hair follicle, glands, sebaceous glands, sweat glands are all located in the dermis. So we're going to turn to the epidermis first. And you should know the tissue of the epidermis. You should already know the tissue of epidermis because the tissue is epithelial tissue. This was our example during the last module of keratinized stratified squamous epithelial tissue. So keratinized stratified squamous epithelial tissue is the tissue found in the epidermis. Because this is epithelial tissue, remember two characteristics of epithelial tissue is that it is avascular. That's what we're always going to have, connective tissue underneath. And the other one is that it is a cellular tissue. So what we're going to have in this tissue is thick layers of cells. Most of the cells are going to be keratinocytes. So these are the most abundant of the cells found in the epidermis. They're called keratinocytes because these are the cells that produce keratin. And we'll talk a little bit more about keratin in a few more PowerPoints. You probably know already keratin is a protein which is made by these keratinocytes that will give the outermost layer of the skin its characteristics. The first layer of cells in the epidermis are stem cells. These are germ cells. These are adult stem cells. The name of these cells is basal cells. And it is a one layer of very, very young cells at the deepest part of the tissue of the keratinized stratified squamous epithelium. Together with the basal cells, interdispersed among the basal cells, we have another population of cells. These are called melanocytes. And melanocytes are pigmented cells that produce a dark pigment that has the function of absorbing UV light. These are pigmented cells, and the pigment is going to give the skin its color. Another category of cells found in the epidermal layer of the skin are Langerhans cells. These are macrophages, which are immune system cells. And finally, Merkel cells are touch receptors found in the epidermis. The dermis is also going to have different types of touch receptors, but the touch receptors found in the epidermis are Merkel cells. So here is a picture of the epidermis. Remember that this is keratinized, stratified, squamous epithelial tissue. And it starts down here. So this entire layer right here is the epidermis, right there. Okay. Which means that this layer underneath here is going to be the dermis. So this is dermis. This is, I should say here, this is also dermis. Okay. All right. So notice that the epidermis has several layers when you look at it closely. First of all, you can see this series of cells right here at the bottom, right there. These cells will be the young basal cells with melanocytes interdispersed among these basal cells. So this is the basal cells and melanocytes found in the deepest layer of the epidermis. As we move up, the cells become lighter in color, perhaps even bigger, and a bit flatter. So these right here are going to be live keratinocytes, which are producing keratin. These are going to be older cells because the young cells are the basal cells. And as the basal cells divide, they're going to send the older cells, they're going to push the older cells toward the more superficial layers. Langerhans cells are also found in these middle layers, roaming around, cleaning the tissue and looking for debris. Now, notice this layer right here. Notice how cells change right here. These cells are again darker, and if you look at it closely, they look like they have granules inside. So now this is going to be another layer of cells within the epidermis, within this stratified tissue that is visible and we can note. And then right here, there's yet another change in these layers of cells that is definitely visible. And then finally, we have a very thick layer of dead cells at the most superficial part of the structure of the tissue. So we're going to be going through these layers and naming them and pointing out the function of the layer and the cells that we find in theirs. Okay, so again, back to melanocytes. We're going to spend a little bit of time talking about melanocytes later on, but melanocytes are found in the lowest layer together with basal cells. So this will be a basal cell. This funny looking cell is going to be the melanocyte. The melanocyte produces a pigment called melanin. It is a brown pigment. About 10 to 25% of the cells in the lower epidermis are going to be melanocytes. And then notice how the cell creates these extensions of the plasm right there. And the extensions go to the higher layers. Okay. And then these little vesicle-looking structures are going to pinch off these extensions. And these vesicles are going to have pigment. So the pigments, these vesicles filled with melanin, are going to be positioned in the upper layers of the skin that are more subject to UV light exposure. The pigment absorbs UV light and prevents UV light damage on the cells of the epidermis. So these cells up here are live cells. They are close enough to the surface to receive UV light damage, and UV light is a mutagen. It can damage DNA. So to prevent damage and mutations of these cells, melanin is going to be present in these vesicles, and it's going to be positioned on top of the nuclei of these cells, preventing damage. So again, this is why melanin is so important, and that's why we're going to talk so much about melanocytes, is because they protect layers of the epidermis from UV light damage. We've mentioned the other population of cells found in the epidermis. These are the Langerhans cells. Epidermal dendritic cells is another name for these cells. These are macrophages, which are the immune system cells. And then we also mentioned the touch receptors found in the epidermis. They're called tactile or Merkel cells. Okay. So now we're going to turn back to the epidermis and to the layers in the epidermis. The layers are called strata, which is another word that means layers. Let's see. And as we go through the stratas, you should be noting things like the cells found in the strata, the characteristics of the strata, the thickness of the strata, and whether the strata is found in thick skin or thin skin or both. Okay. So first of all, let's talk about thin skin and thick skin. And this is actually easy because there's only a few places in the body where we find thick skin. Thick skin is the type of skin found in the palms of the, I'm going to bring you to this PowerPoint here. So thick skin is found in the palms of the hand and the sole of the feet. So palms of hand. So these are the layers, the places where we find calluses. Palms of hand and sole of feet. And those are the places where we find thick skin. You can see here, this would be the thick layer of the outermost part of the epidermis. This is the rest of the epidermis right here. So contrast thickness of this dead layer right here with the thickness of the dead layer in what we call thin skin. So everywhere else we have thin skin, except for the palms of the hand, sole of the feet. Think of the skin that covers the eyelids, for example. That's extremely thin skin. In thin skin, we find a thinner layer of dead cells. So that's essentially the difference between thick skin and thin skin. So it's really not that complicated. And we're going to note which strata is found in each, thick skin and thin skin. Okay, so this is a pretty good drawing of these different layers. The deepest layer, the deepest strata is called the stratum basale or germinativum. It has two names, and this is one layer of cells down here. The cells are basal cells. So these are the germ cells and melanocytes. Melanocytes we mentioned are the cells that produce melanin. Basal cells are germ cells. Germ cells means very, very young cells that are constantly replicating. And as they replicate, they push the older cells towards the surface. So the next layer of cells is the stratum spinosum. And the stratum spinosum is made of these cells which are still alive and they're still fairly young, but they're not germ cells anymore. The next layer up, and by the way, stratum spinosum can be fairly thick. It can be about 10 layers of cells. The next layer is the stratum granulosum, and this is the layer of cells that have begun to flatten. They begin to accumulate keratin, they begin to die, lose their cytoplasm, lose their organelles. So this is a big change that is visible when you look at this layer under the microscope. This is a layer of about three layers of cells. Okay. The next strata is only found in thick skin. So when we mention about thick skin versus thin skin, this is the only strata found in thick skin. All the other strata are found in both thick and thin skin. The stratum lucidum is the first layer of dead cells, and lucidum means clear because these cells are going to look like clear cells. The whole strata will look like a clearing that all of a sudden can be seen above the stratum granulosum. After the stratum lucidum, there is a layer of dead cells, the stratum corneum. Think of the corns of the feet, because the corns of the feet have thick layers of the stratum corneum. These are dead cells. These cells are filled with keratin, and they look more like fibers than they look like cells. So again, we're going to go over these strata. In this particular picture, this is a nice picture. You can see oops, you can see here, down here, the stratum basale, one layer thick. Then you can see above the stratum basale, this layer right here will be the stratum spinosum. You can see the little dots inside these cells. This will be the stratum granulosum. The stratum lucidum is not visible, and you can almost argue this right here will be the stratum lucidum, but I'll show you a picture of the stratum lucidum, and then this will be actually this is thin skin, so forget about the stratum lucidum. And then the layer of stratum corneum. The stratum corneum can be extremely thick, being about 20 or 30 layers of dead cells. A lot of this chapter is just going over these strata. Stratum basale, again, one single row of stem cells and melanocytes. Remember that this is called the stratum germinativum. It has two names. These are cells that are dividing very rapidly. They are young cells, and they're replenishing the epidermis constantly. So it says here that it takes about 40 to 56 days for cells to travel from the basal layer all the way to the top. So this will be the lifespan of these cells, because by the time they get to the top, they're dead. All right. Let's see. The next layer is the stratum spinosum. Spinosum means prickly, and the reason why this is called stratum spinosum is because when these cuts of the skin are prepared to be viewed under a microscope, they put in alcohols, and alcohols shrink the cells. And turns out that the stratum spinosum cells, like all other epithelial cells, are attached to each other. So when they would put the alcohols, the cells will shrink, but they remained attached. And they had this prickly appearance. They look like spines. And that's where the name comes in. So if you think the name is weird, spinosum, well, that's where it comes from. And so the cells that we find in this strata are keratinocytes. These are live keratinocytes. It's still dividing. I should say they're not dividing, but they're still alive, still metabolizing and nucleus. They have organelles. We also find in this layer the immune system cells, Langerhans cells. Keratinocytes are going to be covered by the melanin granules, because these cells, being alive, they need protection from UV light. The thickness of the layer varies between 8 to 10 layers. Superficial to the stratum spinosum is the stratum granulosum. And this is a thinner layer, 3 to 5 thickness. These cells now have flattened. They're in the process of dying. They're losing their organelles, they're losing their nuclei, and they're making abundant keratin. So keratohyalin and lamellated granules accumulate, which gives the cell its name. The next layer is the one that is only found in thick skin. This is a clear layer, because by then the cells have lost their nucleus, they have lost their organelles, and they're filled with keratin. So they look like a clearing above the stratum granulosum. These are just a few rows of cells, maybe 3 to 4 layers, dead, flat, keratin-filled cells. And then finally, the outermost layer is the stratum corneum. The stratum corneum can be very, very thick, and it depends on where we're looking at. It can be as thick as 30 rows of dead cells. In parts of the skin, it may be thinner layers. But in general, this is a pretty thick area. The cells are dead, flat, keratinized, filled with keratin, okay, and represents about 3/4 of the epidermal thickness. Okay, now the keratin itself. So this is the important layer, because this is the layer that is that gives the epidermis its function. Remember that the function of the epidermis, being a stratified tissue, is to protect from mechanical abrasion. So this is a layer that protects from mechanical abrasion. The fact that the cells are dead allows us to shed them if we scratch the skin, for example. Keratin is a waterproof protein, so water cannot go from the surface to deeper areas. So water cannot penetrate the skin. Now, water could leave the skin, so it can evaporate through the skin, but it cannot go from the outside into deeper layers. The keratin prevents water loss. Okay, so even though water does evaporate, because of the presence of keratin, not as much evaporates as it would if keratin was not present. Keratin is also a very powerful barrier against biological attack. Bacteria cannot metabolize keratin, so they cannot eat it. So they cannot live in deep layers in the skin. Bacteria are found on the surface, but they're not found living in deeper layers because they cannot eat keratin. Fungus, however, can. So sometimes the skin does get infected with fungus. But in general, that doesn't happen. The keratin is still a very good barrier. It's also protective against chemicals and physical assault. So again, I cannot emphasize enough, you need to learn the layers, the order of the layers. You should be able to put the layers in order from deep to superficial, or if called to do the opposite, from superficial to deep. Okay, so you should be able to put them in order in whichever direction you're asked to put them in order, from deep to superficial or superficial to deep, and pay attention as to whether asked to the skin mention is thick or thin. If it is thick skin, you need to make sure that the stratum lucidum is there. If it is thin skin, make sure that you don't add stratum lucidum. And this is the PowerPoint I was talking about, where you can actually see the stratum lucidum. And you can see here, put it in blue, this layer right there, this clearing that you see all of a sudden up here above the stratum granulosum, that's the stratum lucidum right there. The stratum granulosum can be seen at this darker layer right there of cells that have the little granules. The stratum spinosum is right there, and the stratum basale is the one layer thick right there at the bottom. Obviously, the stratum corneum is the thicker layer right there. This is the stratum corneum. We don't see a stratum lucidum. This is thin skin. This thin layer right here will be the granulosum, which means that this is the spinosum, and this down here would be the basale. Okay. All right. Again, cannot emphasize enough, there will be questions on the strata and characteristics of the strata. Now, when we talk about skin color, there are different factors that can affect skin color. Circulation, blood circulation will affect it. Especially light skin is affected by blood circulation. Carotene, vitamin D can also affect the color of the skin. The thickness of the skin could also affect its color. Presence of hormones, especially estrogen. And the most important factor is melanin. Okay, so let's talk about things that can affect skin color. Again, the most important factor is melanin. Melanin is a pigment that has anywhere between a reddish to a yellowish, brownish color. It is produced by cells called melanocytes. Melanocytes are found in the stratum basale. We mentioned that already. Okay. Melanin is packaged by melanocytes inside vesicles called melanosomes. So these are the vesicles that are packaging melanin. So you can imagine the little melanocytes putting together these little bags filled with melanin. So the bags are called the melanosomes. Okay. And the pigment is inside the little bags, and they put out into the upper layers of the epidermis, and they position themselves strategically on top of nuclei of cells to protect the DNA in the nuclei from UV damage. They're distributed to keratinocytes in the stratum spinosum. We don't care about the stratum corneum because those cells are dead already. The stratum granulosum, the cells are dying, so it's not the damage that UV light can cause is not going to be relevant. The stratum spinosum, on the other hand, those cells are still alive. And they could mutate and they could become cancer cells and begin to overgrow. So to prevent that from happening, melanin is positioned on top of the DNA of these cells to absorb UV light and prevent and protect them from UV radiation damage. Melanin happens to be a pigment, and the production of melanin is going to be stimulated by UV light. So when we're exposed to sunlight, that's going to stimulate melanocytes into producing more melanin. And that's why the skin tans when we're exposed to sunlight. Now that takes a few days. We don't tan immediately. We might burn immediately, but we're not going to tan immediately. So the process of stimulation takes a few days for it to happen. And again, I cannot emphasize enough, melanin absorbs UV light. So the purpose of melanin is to absorb UV light. So if you're asked a question as to what's the function of melanin, if you answer, it gives a skin color, that's just a partial answer. That's not the ultimate purpose of melanin. That happens to be a side effect of melanin. The purpose of melanin, the function of melanin, is to absorb UV light. Okay, so pay attention to that, and make sure that you understand what the actual function is. It's obviously people that live in tropical areas that are more exposed to UV light, they must have a darker skin, they must have more melanin because they're exposed to more UV light. On the other hand, people that live in areas that they don't have as much sun, like Nordic countries in Europe, for example, they have lighter skin because they don't have the UV light exposure, and so they don't have as much need for the protection. Okay, so let's see. Melanocytes produce melanin, protects from the stratum spinosum from damage by UV light, and prevents mutations, which could lead to skin cancer. All right, protection from sunburns. This is also important to remember. The number of melanocytes is equal, more or less, about among all races. So about 10 to 25% of the cells in the basal layer are going to be melanocytes. So it's not the number of melanocytes that varied among races. What varies is the ability of the melanocytes to produce melanin. So the darker the skin, the more melanin these melanocytes can produce. There are genetic mutations in which the melanocytes are unable to produce melanin. And in those cases, the enzyme that is needed for the production of melanin is lacking, that gene is mutated, and the condition is called albinism. Now, albinism does more than just gives us a hair and a skin that is very, very light. Melanin is also found in the eyes, in the iris of the eyes. So the pigment in the iris will be gone. The eyes typically look reddish because of the lack of pigment. And that can be damaging because the function of melanin is to absorb UV light. And if it is not there, now areas of the skin that shed melanin are now sensitive, exposed to UV light, and they could be that could lead to UV light damage. Okay, so that's the downside, and that's the danger of albinism. So you should know what albinism is and what causes it. Freckles are accumulation of melanin in certain areas of the skin. Okay, so that's where that comes from. So aside from melanin, other factors that could affect skin color would be things, for example, like carotene. And carotene is the storage form of vitamin A. So if you think of carrots, carrots are loaded with vitamin A. Vitamin A, actually they're loaded with carotene, which could be made into carotene and to vitamin A. So carotene is a reddish, I'm sorry, not reddish, well, it could be reddish. It is a orangey reddish kind of vitamin, and sometimes and it is a fat soluble vitamin, which is stored in fat tissue. Now remember, the layer of fat tissue underneath the dermis can store vitamin A, can store carotene, and if we're eating too many carrots, if we're accumulating too much vitamin A, the skin could turn a orangey color. Okay, so there's nothing dangerous about it. Carotene is not toxic. Vitamin A could be toxic in high amounts, but carotene is not. So this happens sometimes to children that eat too many carrots. So that's another factor that can affect the skin color. Hemoglobin, the presence of hemoglobin could change the skin, give it a reddish hue. The conditions may be called erythema, and it's especially noticeable in light skin colors. Now, if hemoglobin could give skin a reddish tone, lack of hemoglobin could give it a bluish tone. So a lack of circulation in the skin turns the skin bluish. The condition is called cyanosis. And it could be a problem with hemoglobin, a lack of blood cells. It could be a heart problem. Sometimes babies are born with heart problems that causes circulatory problems, and these babies are called blue babies because they have a bluish tinge. The areas of the face that especially turn, or the body that especially turn blue, would be the lips. The nails can also turn blue. Okay, so that's what we look for when you're looking for cyanosis. It's a bluish coloration in the lips, the nails, and then depending on the lightness of the skin, you may also see it on the skin. You should know what cyanosis is. Another term that pertains to skin colors is jaundice. Jaundice is a yellow coloration of the skin that is due to an accumulation of a toxin called bilirubin. Bilirubin is an end product of breakdown of red blood cells. So the breakdown of red blood cells happens all the time. Millions of red blood cells get destroyed by the spleen every single day. And what happens to the bilirubin is that it is taken to the liver, and the liver detoxifies the substance, and then we excrete it in feces. We also excrete it in urine, and that's why urine is yellow. Now, if there is a problem with the liver, however, and it cannot detoxify, now bilirubin keeps circulating in the blood, seeps into the skin, and the skin turns yellow. You will also see a yellow coloration on the white of the eyes. So jaundice in an adult indicates liver damage. In a baby, it will indicate excessive destruction of red blood cells due to some type of blood incompatibility with the mother. That's typically what a jaundiced baby is having to deal with some type of blood incompatibility between the mother and the baby. So the baby is born with with excess bilirubin because too many of his red blood cells are being destroyed, and the liver is too young, too small to deal with the excess bilirubin. So you should know what jaundice is, that yellow coloration of the skin, and and conditions that give rise to jaundice that could be indicated by jaundice. Liver disease is typically what we're looking at, or increased red blood cell destruction, especially in babies. So that takes care of the epidermis. Now remember that the lower layer of the skin was called the dermis. This was a connective tissue layer, and this is actually the thicker layer of the skin. And this is a tattoo. When somebody gets a tattoo, the pigment goes into the dermal layer, which is obvious because if it got into the epidermis, it will be eliminated within a matter of a couple of months because that's the layer that's constantly being shed. All right, so a lot of the characteristics of the skin are being given by the dermis. The dermis is strong, it's flexible, it's connective tissue. Because it's connective tissue, all connective tissue, remember, had two components. It had cells and it had matrix. And the matrix had two components, ground substance and protein fibers. And the protein fibers can be collagen, elastic, and reticular fibers. In the case of the dermis, the cells are going to be fibroblasts. Those are the cells that will secrete the protein fibers and the ground substance. Other cells found in the dermis are going to be immune system cells, which makes sense because if there is a damage to the epidermis, now the protective layer is gone, and we better have immune system cells ready to fight off and to prevent a deeper entrance of potential pathogens past the dermis. So that's what we have, the immune system cells right there at the dermis. This is called the second line of defense. The epidermis is the first line of defense. If the first line of defense is damaged, the second line of defense is ready and waiting to prevent an infection. Okay, so those are the kinds of cells that we find in the dermis. Definitely remember the fibroblasts, because those are the cells that are going to create the ground substance and the protein fibers. Now, the dermal layer is made of connective tissue, and is actually made of two different types of connective tissue. The top layer is called the papillary layer, or papillary dermis, and it is made of areolar connective tissue. If you remember last chapter, chapter 4, it spoke of areolar connective tissue as the tissue that was always found underneath the epidermal tissue. So this is the tissue that always support epidermal tissue above. The thickest layer of the dermis, however, is made of dense, irregular connective tissue and makes up what we call the reticular dermis. So the dermis has two parts. It has the papillary layer and it has the reticular layer. The papillary layer is very thin, just up here at the very top. Okay. The reticular layer is the thickest part, made of dense, irregular connective tissue, while the papillary layer is made of areolar connective tissue. So let me talk about the papillary layer first. And let me just see something. Yeah. So let me erase these because I want to show you something. The reason why this is called papillary. Papillary is a term that we use a lot in biology. So this is not going to be the first time you hear this term. Papillary means wavy, like a papilla. So the papilla is at these waves right here that you see there. That's a papilla. Okay, that's why this layer is called papillary, is because we see these waves right there. Okay. Now, the layers of the dermis are two, the papillary layer and the reticular layer. These are called dermal layers, okay, or layers of the dermis. When it comes to the test, be careful. Read questions carefully. If a question asks about dermal layers, then the question pertains to one of these two layers, papillary layer or reticular layer. If a question asks about epidermal strata, now what the question is asking about the different strata of the epidermis. The stratum basale, the stratum germinativum, the stratum granulosum, the stratum lucidum, the stratum corneum. Those are epidermal strata. So if a question asks in which epidermal strata do we find Langerhans cells, for example, and one of the options is papillary layer, well, don't choose that option. That option is obviously not epidermal strata. Okay, that is dermal layer. If a question asks which dermal layer has areolar connective tissue, don't choose epidermal strata, even if it's one of the options, because we're not talking about the epidermis. The question is asking about the dermal layers. So don't let the terms confuse you. Slow down, read the questions, and make sure you understand what the question is asking. So that's just test-taking tips here. Okay, so going back to the dermal layers. First layer is the papillary layer, and the papillary layer is made of areolar connective tissue. This is what areolar connective tissue looks like. And this is a tissue that has all three types of fibers: collagen fibers, elastic fibers, reticular fibers. It has blood vessels, little capillaries that feed the epidermis above. Okay. It has what was called the dermal papillae, and the dermal papilla are these little up and down ridges that I mentioned right there. Meissner's corpuscles are also found in the papillary layer. Meissner's corpuscles were touch receptors, and these were touch receptors for light touch. And also found in this layer. Let me show you what we mean by papillary ridges right here. So this is a picture that is showing you dermal papillary ridges. This layer up here, A, is the epidermis. Okay, that's the epidermis. Okay, and down here is the dermis, and actually is the first layer of the dermis, so that's the papillary layer. What the picture has done is has separated the two layers. Now you can see these ridges going up and down of the papillary dermis, and you can see how the ridges are followed by a complementary shape on the epidermis. So they kind of interlock with each other, and that's what creates the fingerprints or friction ridges. Skin markings are created by these interlocking of the dermopapillary ridges and the epidermal ridges. Okay, so I just wanted to point out what that meant. Okay, so that takes care of the papillary dermis. The reticular layer of the dermis is the thickest layer. About 80% of the dermis will be this reticular layer. It is made of dense, irregular connective tissue, lots of collagen fibers. You can see the huge bundles of collagen fibers, elastic fibers, and it is the elastic fibers that give the skin its ability to recoil. So you can take skin and stretch it and then let it go, and goes back to its original shape, thanks to the elastic layer fibers. Now the elastic fibers may break, and then we have stretch marks. Okay, so these thick layers of collagen fibers create bundles or arrangements that that are found in different directions all over the body. This is what we call cleavage lines. So cleavage lines refer to these bundles of collagen fibers found on the dermis all over the body. And plastic surgeons study cleavage lines because these lines are going in mostly different directions depending on the part of the body that you're looking at. So when surgeons cut the skin, they try to make the cuts parallel to the cleavage lines in order to minimize scarring. If you cut perpendicular to the cleavage lines, the skin will have a harder time, that dermal layers will have a harder time repairing, and you'll end up with a bad scar. So you should know what cleavage lines are and why they're important. Okay, so that takes care of the dermis. If there are no questions, okay, is the papillary dermis the same as lamina? Oh, good question. Yes, it is. Yeah, the papillary layer is the lamina propria. Probably, technically speaking, the lamina propria may be the top layer of the papillary dermis. But yeah, it is exactly the same thing. The lamina propria is made of areolar connective tissue, and it's that layer of papillary dermis. And actually, let me go back here. Keep in mind, the lamina propria is found on any tissue, any epithelial tissue. Underneath it will have areolar connective tissue and lamina propria. In the skin, that lamina propria would be the papillary layer. So the answer to your question is yes, but only in the skin. If we're looking at the intestines, for example, we're not going to call that the areolar, the papillary dermis. We're going to call that, actually has a different name. We call it the mucosa layer. But we don't see that this semester. So the answer to your question is yes, only in the skin, however. Okay, so hopefully that clears it up. Okay, no problem. All right, so now we're going to turn to glands, and we're going to talk about, especially about sweat glands and oil glands. Those are the two things, the two types of glands we're going to talk about. These are going to be other structures, other organs in the skin. We'll talk a little bit about hairs and hair follicles. We're going to barely mention nails. So when it comes to glands, we'll talk about sweat glands and oil glands. Another word for sweat gland, another name for these sweat glands are sudoriferous glands. You should know there are two types of sudoriferous glands. We have eccrine glands, which are also called merocrine. And these are the sweat glands that we're more familiar with. They are all over the body, but especially in the palms of the hands, sole of the feet, and they are the ones, the forehead. They're the ones that secrete the watery type of sweat. So these are the kinds of glands that when we think of sweat glands, we're thinking of these types of glands. The other type of sweat glands are called apocrine sweat glands, and they're found only in certain parts of the skin, under the arms, in the genital area. Their secretion is a thicker secretion. It's a mixture of oil and proteins, and is typically smelly. These kinds of glands are stimulated by sex hormones, estrogen and testosterone, which begin to be secreted at puberty. So these glands become active, especially at puberty. So you should be able to distinguish between eccrine or merocrine glands, which secrete mostly a watery type of sweat. The function of that sweat will be thermoregulation. This is what creates what we call sensible perspiration. There's also some antibacterial and dermicidal properties in sweat. And this is a depiction, a picture of these kinds of eccrine or merocrine sweat glands. The other type of sweat gland are the apocrine sweat glands. And these types of sudoriferous glands produce a substance that has sebum or oil intermixed with proteins. This is a smelly kind of sweat and functions as at puberty. Okay. Another form of sweat gland are the ceruminous or waxy glands found in the external ear canal. So they secrete the earwax. Mammary glands are also modified sweat glands. The other types of glands found in the skin are sebaceous or oil glands. They're distributed all over the body except palms of the hand and sole of the feet. They develop, we find them developed with hair follicles. Some sweat glands, however, are by themselves without any type of hair follicle around. They also become more active at puberty due to the actions of sex hormones. And that's what we end up with acne at puberty. Sebaceous glands secrete sebum. Sebum is an oily holocrine secretion, meaning that the entire cell is being secreted. It has a bactericidal property because it has acidic, it has fatty acids. Its function in softening the skin and the hair. So the sebaceous glands that we mentioned typically are the ones associated with hair follicles that you can see right there. So you should distinguish between sebaceous glands and sudoriferous glands, and remember there's two types of sudoriferous glands that we mentioned. When it comes to hair, we're going to go very lightly on hair, just mention a few characteristics. Function is to protection against trauma, heat loss, sunlight. Also alerts of the presence of insects on the skin. That's, each little hair has a little nerve ending wrapped around it. So we can actually feel every individual hair. Hair is distributed all over the body. There are a few areas that do not have hair: the lips, nipples, portions of the external genitalia, palms of the hands, soles of the feet. Hair is made of keratinized cells. So this is a combination of epithelial tissue with obviously connective tissue underneath to keep it alive. The epithelial cells found in the hair are interdispersed with melanocytes, and that's why hair has a pigment. It's because there are melanocytes found there too that produce melanin. The melanin may be a little bit different than the one found in the skin. It's a bit tougher, and there is more of a variety of colors when it comes to the melanin produced by the melanocytes in hair. And this is a picture here of a hair follicle, a cross-section of a hair follicle. Or hair, actually. The follicle is this area down over here, and you can see the thick layer of epithelial cells that make up hair. And this layer right there will be the basal cells where the melanocytes will be found. Okay. Let's see. Yeah, you know that hair bulb is the area, the expanded area at the bottom, at the hair root. Let's see, we talked about sensory endings at each hair so that we can feel each one of them. You know, from lab, there's a little smooth muscle attached to the hair called the erectropili muscle that gives us goosebumps. Know that there's a sebaceous gland associated with each hair. Okay, so there's the little sebaceous gland right there. All of these, we already saw it in lab. Let's see. Yeah, hair growth in phases, and there's always a resting phase of a few months in between. Alopecia is the thinning of the hair that occurs in both sexes after age 40, and that's due to the hair follicles dying. So hair follicles, eventually lose their hair and they regrow a new hair. But that whole process begins to slow down, and eventually some of the hair follicles stop working altogether, and that gives us the thinning of the hair. True baldness is determined genetically, and is typically due to a excess of testosterone that is turned into DHT, which attacks hair follicles. Okay. When it comes to nails, the only thing you need to know, this is again keratinized tissue. So dead cells, the live cells are found underneath the cuticle. Those are the live epithelial cells that are constantly dividing, and the dead cells are keratinized cells. Dead keratinized cells are pushed to the surface, and that's what we call the nails. So hair and nails on the surface are just dead tissue. All right, so we're going to go now over the functions, if there are no questions. The next PowerPoints go over the functions of the skin, of the integumentary system, I should say, not just the skin. And typically the first function we think about when we think of the integumentary system is protection. Protection from different attacks, mechanical attacks, biological barrier, as a chemical barrier, protects from excessive, low pH, for example. So protection is the first thing. The second function we have talked about in chapter 1 a lot is thermoregulation, regulation of body temperature. Sweat is going to cool us off under hyperthermia. Under low temperatures, the blood vessels in the skin are going to constrict, preventing excessive heating loss. When the temperature is high, the blood vessels of the skin dilate to release heat. So this little PowerPoint here is again showing you when the temperature, actually this is normal blood temperature, normal blood flow under normal temperature. Under heat, these blood vessels are going to dilate, and blood will be diverted from the center of the body to the skin to lose the excess heat. And do review yet one more time thermoregulation and the role of the skin in thermoregulation. High temperatures, high temperatures, blood vessels are going to dilate to lose heat. Blood vessels of the skin will dilate, and sweat glands will secrete sweat to cool off the body temperature. Under low temperatures, blood vessels of the skin will shrink to prevent heat loss. Skeletal muscle begins to shiver, to move, and that brings the temperature down. So review thermoregulation again. Another function of the integumentary system is the detection of senses, of general senses. Changes in temperature, changes in touch, pain receptors are all found in the skin. There's a metabolic function of the skin. This is important, and we'll see it again in next chapter 6. The synthesis of vitamin D begins in the skin. And vitamin D has several functions. It allows us to get calcium from foods that have calcium in the intestines. It also seems to have a protective function from cancer, from carcinogens. So vitamin D is made in the skin, or at least the first steps occur in the skin. It is a long process, but the first step happens in the skin. And actually, we need UV light, we need sunlight for that process to start. So UV light, sunlight, has two sides of UV light. On the one hand, it might damage the skin, and so we need pigment to protect us from excess UV light. On the same token, we need it in order to make vitamin D. So we need some exposure in order to synthesize vitamin D, but too much exposure could be damaging. So that's the tricky thing about UV light. Another function of the integumentary system is a blood reservoir. At any given time, there's at least about 5% of the blood will be found in the skin, and that's why peripheral skin vessels dilate or shrink depending on what the body needs. The skin is also an excretory organ. We get rid of waste through the skin, through sweat. Salt, nitrogenous waste is expelled also through the skin, not just through the urinary system. Okay. Last PowerPoint I'm going to talk about is skin cancer and burns. And when it comes to skin cancer, you should know the risk factors will be overexposure to UV light or to irritants. Irritants could be chemicals, for example. There are three major types of skin cancer: basal cell carcinoma, squamous cell carcinoma, and melanomas. Basal cell carcinomas these types are named after the cells that cause the cancer. In the case of basal cell carcinomas, the cells that are causing the cancer are the cells in the stratum basale, which begin to grow uncontrollably. This is a fairly common type of cancer, but it is fairly easy to treat. It is typically treated with surgery, just removing the lesion. And this is what the lesion looks like right there. You typically find it in places that stick out, like the nose or the ears, because those are the places that will be exposed to UV light the most. The second most common type of cancer is squamous cell carcinoma. In this case, the cancer is made by these squamous cells in the stratum spinosum. These are the keratinocytes. Remember, the tissue is stratified squamous epithelial tissue. So keratinocytes in the stratum spinosum may get mutated, they begin to grow too much, and they produce these cancerous lesions. This is a lesion that is typically, again, treated with surgery by removing it, and then there may also be some radiation therapy. Fairly easy to treat, especially if caught in time. It doesn't really represent much of a problem. The lesion itself looks like a wound, an open wound that doesn't heal, and it may just remain for months and months. So it's a very characteristic type of lesion. The most serious type of skin cancer is called melanoma. The cells involved are melanocytes. And the reason why this is such a serious type of cancer is because melanocytes are resistant to radiation by nature. They are filled with a pigment that absorbs radiation. So radiation therapy is not useful in this case. Chemotherapy is not very useful either because the cells very quickly become resistant to chemotherapy. The best course of action is early detection. Detecting it early and removing it through surgery. That is the best way to get rid of this type of cancer. It is metastatic. It very quickly begins to spread. So it's a very serious type of cancer. So it is crucial to be able to identify melanoma lesions. What does the melanoma looks like on the skin? To identify it, dermatologists have created what they call the ABCD rule. And the ABCD rule describes the melanoma lesion right here. So A stands for asymmetric. So the lesion is not the same. If you were to cut the lesion, the two halves are not the same. So in the case of a melanoma lesion, if you were to cut them, the two sides are not going to look the same. Okay, so this is the A in the ABCD description. The B stands for borders. So asymmetry, then borders, the lesion exhibits indentation. So the edges are not as smooth. Okay, so if you look here, the edges of the lesion are not as smooth. Look at a pigment, at a little freckle in your skin, and notice that the borders are smooth. So that's a benign lesion. And melanoma doesn't have smooth, even edges. It has uneven edges. So that's what the B stands for. So asymmetry, borders that have indentations. C is for color. The lesion typically has more than one color. So it may be blue and dark brown, or black, sometimes reddish. So again, take a look at the picture here, and you can see how the center of the lesion has a lighter color than the edges, which are darker. And the D stands for diameter. This is fairly big, about 6 millimeters, or the size of a pencil eraser. This is typically not a little pinpoint lesion, but it's a bigger lesion. You should definitely know what we mean by the ABCD rule that describes melanoma lesions. So you should definitely be able to know that. describe a melanoma. Next thing talks about burns. And burns are tissue damage of the skin due to heat or chemicals. The danger with burns is that they damage the protective layer on the outside of our bodies, and that leaves us subject to dehydration, to losing electrolytes, which could lead to damage in the kidneys or blood pressure becoming very, very low. We'll talk about the 3 different types of burns, but before that, let me talk about what's called the rule of 9s. So the way that a burn is assessed when it happens throughout the body, and they say, okay, 50% of the body has been burned. Well, they assess that because they have taken the body, and clinicians have divided the body into 9% regions. And by looking at how much of each region has been burned, they can quickly assess the percentage of the body that has been burned. So, for example, the front of the head is 4.5%. The back will be the other 4.5%. So the head itself will be 9%. The limbs, the upper limbs are going to be 4.5%. So anterior and posterior of the upper limbs will represent 9% each, which both of them will be 18%. The trunk will be 18%, anterior 80% on the posterior side, so that will be 36%. The perineum represents 1%, and then the lower limbs is 9% anterior, 9% posterior, 18% for each of the limbs. The 2 together will be 36%. So that would be 100%. Okay. So you should know what we mean by the rules of 9. Now, the burns are categorized according to the layers of the skin that have been burned. A first-degree burn is one in which only the top layer, the epidermis, has been damaged. The burn is typically localized, and what we see is redness, edema, so a little bit of swelling, and pain. So this picture right here, the fingers on the picture exhibit a first-degree burn, just the redness and the swelling. The second-degree burn, the difference between a second-degree burn and a first-degree burn is that in the second-degree burn, now we have some damage of the dermal layer. And what happens is that the epidermal and dermal layer separate, and a blister or an accumulation of fluid, which creates a blister, appears between these 2 layers. So the characteristic of a second-degree burn is the immediate appearance of a blister. So a second-degree burn is going to have all the characteristics of a first-degree burn plus a blister. So it's going to have the redness, the edema, the pain, and a blister. And the key is that the blister appears immediately because there's that separation between epidermal and dermal layers. So in the picture right here, the palms of the hand have a blister. So that is showing you a second-degree burn. Third-degree burn is the most serious kind of burn in which all 3 layers of the skin, or 2 layers, and sometimes even deeper into the adipose tissue layer, have been burned. The color may change depending on the degree of the burn, on the type of burn, what caused the burn. It could be black, it could be cherry red, it could be gray or white. There is no initially there's no edema or pain because the nerves have been destroyed. Typically, these kinds of burns require surgery, skin grafting, and they are critical. So there's a little picture here of a third-degree burn, and it gives you the seriousness of the burn. And a severe burn will be critical if more than 25% of the body has second-degree burns. Okay, now remember that the entire lower limb is 18%. So a second-degree burn that involves more than just the entire limb, one lower limb plus a few more layers, that will be critical. Okay, so more than 25-degree burn, a second-degree burn is critical. More than 10% of the body with a third-degree burn is critical. Okay, if the burn happens in the hands, the face, the feet, a third-degree burn in those areas will be considered critical. The reason why, again, a burn is dangerous why it's critical is because, going back to this first PowerPoint, is because it subjects the body to infection. We have destroyed the protective layer of the skin, so we're going to get infections, dehydration, electrolyte imbalance, and now we have not only a potential infection, but we may have renal shutdown and circulatory shock, which means low blood pressure, which is extremely dangerous. So those are the dangers of a critical burn. And you should know that information. You should be able to describe first, second, or third degree burns. When are they critical? And what is so critical about a burn, the danger of a burn? Okay. We don't look at the development of the skin. We do mention, however, what happens to the skin as we age a little bit. Epidermal replacement becomes slower. The epidermis becomes thinner, sometimes drier, because again, sebaceous glands may not be working as much. The subcutaneous fat layer is thinner also. The elasticity decreases, wrinkles appear. We have a harder time keeping thermoregulating. So the patient may be colder quicker and easier, or overheat easier. There's an increased risk of cancer, sometimes due to accumulated damage over the years, and also due to the decrease in the number of melanocytes. So there's less melanin to protect us, and there are less of immune system cells to protect us. So you should know what happens to the skin, the integumentary system, as we age. Okay, so that takes care of the integumentary system. Hopefully this has been helpful.